Good morning, everybody. My name is Chris Meiers, and I'm one of the researchers in the Genome Engineering Shared Resource, which is one of the shared resources of the Masonic Cancer Center. And I'm going to talk a little bit about the background of kind of where we're at with current day genome engineering, as well as kind of some of the services we provide, some examples of those, and kind of some of our basic processes in the GESR. So let's get started. Oh yeah, GESR, an affordable resource for genome edited cell lines, iPSC and mice is the title. We get into a little bit of all of that. So yeah, again, starting with a little bit of background about targeted nucleases. So it's been an evolution over the years. So starting with engineered meganucleases in 1994, we moved forward through zinc finger nucleases, talons, and today we're with CRISPR-Cas9. And basically CRISPR-Cas9 is the de facto in most cases, as it is very specific and we're getting better and better at it every year. The specificity and the efficiency is going up as we're kind of refining a lot of these methods. But the thing that kind of really makes it targeted is the ability to use guide RNAs to get to a specific sequence in the cells. So the basic process of using CRISPR-Cas9 systems is inducing a double-stranded break at a specific point in the genome. So we are causing what is normally like a pretty drastic event. And so there are two methods that the cell primarily uses to repair those breaks. One of them is NHEJ or non-homologous end joining, and the other one is homologous recombination or HR. Non-homologous end joining is error prone, and so what it is doing is trying to stitch together those breaks really quickly. And so a lot of times there are bases that are lost or inserted at that site. And so the simplest way that we use to knock out a gene is by doing a cut very near the start codon of a protein, such that the start codon is interrupted with a frame shift mutation. And so it's actually very efficient for doing that. And we have gotten really efficient guide RNAs nowadays such that we can get pretty efficient knockout very quickly. The kind of more complicated scenario is using homologous recombination to do the repair. Homologous recombination happens at a slower rate, but when you induce a double-stranded break, you get a little bit of an increased homologous recombination, but it is definitely not the more efficient and prevalent repair mechanism, such that NHEJ is by far preferred. And so you're trying to go for the less efficient method, but it is the best way to get a clean repair. So in that case, it uses a template, which is usually the sister chromatid, so it has the exact same sequence. And so it uses that as a template to build on. However, it's also a way to introduce mutations that we would desire. So you can use that homology to kind of sneak in something that you like. So in a lot of cases, if you have like a single point mutation that is a disease phenotype, then you want to have pretty much all of the same wild type sequence and then just one switch there. And so you get pretty much everything the same except for one little switch. And so homologous recombination is a good way to do some error correction, or if you want to introduce some sort of artificial construct in there, that's another way of doing it. And now I'm going to talk a little bit about how we get these editing reagents into cells. So the two primary methods that we use are electroporation and liposuction. Electroporation is probably the one we use the most. It's pretty simple. You just introduce an electric field, and that causes a permeability of the membrane, and it allows reagents to enter the cell, and then those pores seal up. and then the cell does its thing. Similarly, liposuction, you're introducing the reagents, but they're encased in a lipid complex, and then those are delivered into the cell. And what we're usually delivering is either a Cas9 protein or mRNA, the guide RNA, and then if we have some sort of donor template, that's primarily what we're doing. But there may also be some other compounds or different complexes that we might deliver depending on what the project is. But this is the primary means of doing so. One thing that we've gotten into more recently with the delivery of new instruments is using the MaxQuant Tito to sort cells using fluorescence. So this system is a closed system such that if you load everything in a sterile environment, everything remains sterile throughout. So what it does is, or at least the way we're using it, there are many ways to use it, is that we introduce fluorescence, and we're usually using GFP in the form of either an mRNA or a plasmid, and that is used as a gauge for editing. So basically it's delivered along with all of the editing reagents. And then using this MaxQuant Tito system, you can sort for the high fluorescence. So kind of as a gauge that those cells, if they've gotten a high enough amount of GFP, they've probably also gotten a high amount of those editing reagents. And then using this system, you can sort for those higher expressors for hopefully a higher outcome for that. So if you're trying to just get rapid enrichment of NHEJ, you can get it up to 39-fold of what it originally was. So in a lot of cases, we're getting pretty efficient knockout with just standard NHEJ experiments, knocking out that start codon. However, in a lot of cases, we're also dealing with, for whatever reason, sometimes it's template dependent, sometimes it's donor dependent, but we might have low efficiency of that knockout or knock-in, depending on what we're dealing with. So it's a way of trying to kind of juice those editing events that we want. So in some cases, the original editing event might have only been 5% of what you want. And so using this, you can upgrade it a lot. In one case I saw, I was getting 5% around there, and using some initial experiments, I was getting around 25%. So it doesn't sound like a lot, but when you're going through 96 well plates to subclone, 5% to 25% is a big difference, and it really speeds up the time. And so that's one of the ways that we're kind of using to try and improve our processes and try and get these projects out to people sooner. Oh yeah, and one of the ways this system works a little bit differently than your standard flow sorter. So usually there's some voltage involved. In this case, instead of voltage gate, it's using these detection lasers that are detecting the different fluorescence, and instead it actually just uses a physical valve. So based on the timing of the laser and the cells moving through the flow cell, it will open the valve and sort into a positive chamber. And it's kind of a nice thing, because if you say you're running through and you're not getting what you want, it has a negative sort chamber, so you can always pull back and resort later. So it's a little bit nicer just because your recovery is a little better, since you're not having nearly as much of a stressful environment on the cells. So kind of an example of this is this. This is a method that was used using FACS instead. So starting with like a 14.5% level of indels and insertions and deletions in the population, you can use this level of fluorescence on this method to really juice up that editing. So using the GFP expression as a indicator of editing, You can sort for just those highly enriched edited populations. So you can go from a 14.5% in the total population, and you can see as it goes up to the top pool, you're getting close to 60%. And again, that makes it just so much better when you're trying to sort through all of these different clones. And this was actually using a method that used a reagent that looked for mismatches from the wild type sequence. So it kind of used a fluorescent indicator for those events where there are insertions or deletions. So talking a little bit more about kind of what we do day to day, one of the things that we have to do all the time is quantify all of the edits that we are doing. So the simplest way, if we're doing just a simple knockout, is to use TIDE, which is tracking of indels by decomposition assay. And all you have to do is you do the editing event. So you use your guide RNA to induce those insertions and deletions. You do PCR for the wild type and the edited cells, and then you compare them using this TIDE method. So you just need a 300 to 400 base pair amplicon and send it in for sequencing, get those Sanger reactions back, and then input the Sanger files into TIDE, and then provide the guide RNA sequence. And it outputs this pretty nice little chart. So you get, there are some cases where you do get actual like good repair where there are no insertions or deletions. That's usually a portion of what you have. But then you also get some where you get an insertion of +1, a deletion of -1, deletion of -5. And it does seem to, it varies, from experiment to experiment and from guide RNA to guide RNA, how efficient those are. But you do generally get a pretty good outcome for most guide RNAs. But we also test multiple so that we get what the best and most efficient way so that nobody is wasting anybody's time sorting through clones when you don't have to, when there's usually a better guide RNA out there. One of the other things, and that's kind of a portion of this, is our project reports. So when we complete a project all the way through, we kind of give an outline of what we did. We give reagent details on how the editing was done, along with methods, experimental conditions, and then we provide project data from all of the different data analyses that were performed here. So for example, a simple knockout clone, we would probably have the editing methods that we use. We would have the bulk data. So from an initial edit, that kind of TIDE analysis, or depending on the project, whatever analyses indicating the level of editing in that first experiment. And then we would have the methods that we used for screening clones. So PCR, maybe restriction digest, depending on the project. And then a TIDE analysis of the isolated knockout clones. So for a standard knockout project, if we have a pretty good efficient knockout, we can usually get 3 to 4 clones pretty easily. And so we'd have analyses for all of those, and we'd deliver those, of course. So talking about some kind of more newer methods, we're going to talk about base editors and prime editing, and how those are used and how they're kind of becoming a very useful tool in the marketplace. So the original base editor was developed using a dead Cas9 linked to a cytidine deaminase. And so what that would do is convert the cytosine to a uracil, and then the host machinery, recognizing the uracil, would remove it. And so you could get a transition to a thymine from that. But then doing some kind of more improved optimization of this system led to them using this uracil DNA glycosylase, because a lot of times what would happen is the DNA repair, base excision repair, would come back in and repair it back to the original cytosine. And so that was one way that they were able to improve it. And then they improved it further by adding a nickase Cas9 to mark the edited strand as the parental strand. And this base editing system has a very nice specificity. So around a 5 base pair window, in which you get really high editing. So it's pretty useful under very specific circumstances, obviously. But in a lot of cases, you're able to get pretty efficient editing that way. Okay, there we go. So that's the cytosine base editor and then the adenine base editor. So C to T and then A to G are these two different things. And they're similar. They both do this 5 base pair window. And they, more importantly, they have a really low level of indel formation compared to standard Cas9. So it makes it much more useful when we're talking about doing actual editing experiments in live people and cell therapies and things like that. Because obviously, if we're trying to correct a very specific point mutation or a very specific mutation in a sensitive area, you do not want to introduce any further complications to the scenario. So trying to get these edits very specifically in the way that we want them is what we're driving towards for various therapies. So this is a nice way of doing that. Obviously, it doesn't fit all scenarios, but it can be very useful in those contexts. So this is something that people are looking at a lot in terms of trying to improve efficiencies of these and trying to reduce any potential off-target events that might happen. But with these optimization methods, we've really kind of honed these in quite a bit over the years. Another one is using prime editing. So this is Cas9 fused to a reverse transcriptase. And this is prime editor 3, the third iteration that we're talking about here. So what we do is we have a Cas9 nickase that nicks the DNA strand, so it doesn't induce a double-stranded break. And it uses the single-stranded DNA as a binding template for a PEG RNA or a prime editor guide RNA. And that binds to the single-stranded DNA. However, it's not just homology to that single-stranded DNA. It also has its own changes that we're introducing using that PEG RNA. And so it forms this DNA-RNA hybrid. And using that, you can introduce up to 40 base pairs that are not originally in that parental strand. And it can also delete 80 base pairs using that as a repair template because the reverse transcriptase produces DNA that is used as a repair template for homologous recombination. And then that is introduced into the cell using DNA repair mechanisms. So it can be a nice way of getting a lot of DNA in there. Obviously, the amount of work to design good PEG RNAs is a little bit more complicated in the current atmosphere, but I'm sure it'll get much more efficient over the years, just like standard Cas9 editing has for guide RNAs and everything. So talking a little bit about what we do and some of the projects that we've done, I'm going to kind of talk about our general flow for our projects. So we obviously want to get whatever gene you're interested in knocked out. That's the goal. And so for a simple knockout, you're trying to find the exon that makes the most sense to get rid of. A lot of times it's an early exon, but obviously there are a lot of different scenarios where there's different splicing schemes and things like that. So we're going to do a little bit of research to find the different transcript variants, looking at important domains that are known about whatever your gene of interest is, and then finding the potential guide RNA sequences that target those areas. There's usually multiple options, and you can use multiple different websites that have a good database for finding those guide RNAs to target the specific sequences. Typically, we're finding a couple of different options to find the most efficient so that we are not spending too much time looking for fairly rare clones, whereas we can usually find pretty efficient guides quickly. But what you're looking for is probably a truncation in most cases, such that, yeah, you're interrupting that start codon so that the whole protein isn't produced. However, it can be different depending on whatever your protein scenario is. And if there is something that you know about it that we don't, or something that is newer in the literature, obviously we take that input as we're designing the project. But if it's something that's a little bit less known, then we'll do some work and try and find the best scenario for that. What we then do is we do electroporation using the most efficient guide that we found. We then plate those cells, grow them up, and use tide to determine how efficient our experiment was. If we have efficient knockout, then we go on to subclone. So usually for 96 well plates, and if we have really efficient knockout, maybe even less. And so then we find multiple single cell clones and we genotype them just using PCR and then looking at tide. And that's usually done from a larger well. If you have really efficiently growing cells, maybe we do it out of the 96, but it's usually a little bit safer to go to the 24 wells. And then, yeah, we determine the genotypes of those individual clones. So you will get a blend of different options, obviously. In some cases, you'll get a nice clean indel. You'll get it like a -1 knockout or whatever at the guide site. Although you might also get some wild type clones, as I mentioned, so those won't be very useful, although they might be useful as a control of editing, so we usually keep those. And there might be some where there is an insertion or deletion, but it does not affect the start codon. So, +3, -3 that are, before or after that start codon are not very useful in most cases. So we're sorting through those clones to find the ones that are knocked out efficiently. So an example of this, this is from a FANCD2 knockout that we did of exon 11. And using electroporation, Cas9 mRNA, and a guide RNA, we produced this knockout, so it had 80.7% efficiency. So in this case, actually, the wild type was by far the minority compared to a lot of these indels. And you can see there's a preference towards a -2. So it isn't very surprising that we're able to isolate a -2 homozygous knockout. So we just took that bulk edited population, and I know that we had multiple clones for this experiment, but just showing the one for clone 8 that we found. And so that was just a FANCD2 knockout in BJH TERT cells, but relatively efficient knockout, so pretty quick project to do. If we have a fast growing cell type, these can be done very quickly. One that's a little bit more complicated is a conditional knockout. So in this case, what we did is we used a plasmid that was packaged into AAV and induced a break at 2 sites on the 5' and 3' sides of the gene of interest. In this case, it was BRCA2. And then using AAV after the electroporation, the AAV provided a single-stranded donor, and then using homologous recombination, we're able to introduce LOX-P sites on either side. So those LOX-P sites, when you use Cre-recombinase, it basically loops out anything in between those 2 LOX-P sites and kind of fuses back together such that you get a functional deletion of the whole, whatever's in between those, in this case, BRCA2. So you can do that using Cre-recombinase, but then you can still have that cell line that has the functional, so you can look at before and after the knockout without having to have a knockout cell line in perpetuity. So it can be useful for looking at very specific regulation of whatever mechanism that you're looking at. These are a little bit more tough. As I said, the homologous recombination happens at a much slower rate than NHEJ. So it takes a lot longer to get one where you have a nice clean integration. You can get instances where there's homologous recombination on one allele, but on the other you get something else. If you're lucky, you get a wild type and you can re-edit. But in a lot of cases, you get insertions or deletions if you're using standard Cas9. And so those just take a lot longer to find the clones that you want. Involves a lot of genotyping because you're checking both sides of a pretty, in this case, a large gene. Most genes are pretty big, so it takes a lot of PCR to find those. So it takes time, but it can be obviously useful for you to have something like this and to not take the time yourselves to do it, which is why we're here. One thing that we do that as people have, as we've developed as a field to get more efficient knockout is just a population knockout. So if you have the time and we have really efficient knockout, this can be a good way to save a little bit of money and to get what you want and potentially get more clones that you want to kind of investigate. So we have these really efficient guides, and since we have like 80% knockout in some cases, you have a lot of good clones to choose from, such that subcloning is not as time consuming as it used to be. And so what we do is we do the knockout experiment. We grow up the cells, we genotype them, we genotype the population using tide analyses, and then we provide a report of how efficient it was and provide the cells. For that, a highly edited population, it's only $2,500. You can do the subcloning yourself, and then you can subclone as many clones as you want to from that edited population. And a newer thing that we've done a little bit here is overexpression cell lines. So this is a little bit less complicated since we're not using Cas9. You can use transposons or lentivirus to deliver whatever protein or gene of interest into your cells. And it can provide, for transposons, high level of protein expression without the need to make viral particles. And with lentivirus, you can deliver whatever you need without the need to do electroporation. So depending on your cell type, depending on your gene, this may be very useful because you can use any cDNA. And you don't have to worry about insertion into a specific site because they insert all over the place. So if you're just looking at a protein, or if you have a tricky cell line that doesn't react well to electroporation, this might be a good option. And if we're using selection as part of that, then it can be really efficient because we can just use whatever selection drug and just isolate individual clones. And usually we have pretty efficient ones. And especially using Tito now, we can use antibodies to mark those cells and flow sort them and get them really efficiently. Really efficient clones for those transposons or the lentivirus. And talking about mice, which is something we've done a lot with the MGL. More recently, we've done a lot of conditional knockouts, but there's a lot of, if we're doing a standard knockout, it's even easier. But what we're usually doing is using RAAV and Cas9 in early embryos. So this is very efficient. We usually get pups that are genotyped within a couple of months. And with less time needed for breeding, you can really expedite timelines on designing your mice. So for example, we did one for Septin4 conditional knockout mice. And within the first round of editing, we had 3 mice that had correct integration by PCR. And that's something we also provide is, depending on what the needs of your laboratory are, it can be just faster for us to do testing for you. We have a pretty efficient method for testing. So for the actual mouse knockout process with the MGL, it's about $4,000, but then additional costs for genotyping apply. But it can be a pretty efficient way of doing this because you can get those mice in the first round, breed them, and get your conditional knockout pretty efficiently versus older methods. Not too complicated. I believe you're usually using a wild type mouse and an edited mouse to get what you want in the end, but that's something to talk about the MGL about more so. But we do a lot of editing work with them. We do design and then do validation prior to the editing such that we know that we have really efficient guides for cutting and for those experiments. So talking about some cell lines we've dealt with. So we have a variety of different cell types that we've engineered. So you can see here we've dealt with some human cell lines. We are dealing with mouse cell lines. So NIH3T3 is our go-to for doing validation of guides for these mouse editing experiments. We've worked with dog, we've worked with rat, and obviously we're pretty proficient in human cancer cell lines. We have started offering iPSC engineering, and we're housed now within the Moriarty and Weber labs, or just adjacent to. So they have a wealth of resources that we're using to offer this new source of iPSC engineered cells. So if that's something you're interested in, you can talk to us about it. We haven't actually done one yet, but we're definitely psyched to try it out because I think it'll be a pretty interesting direction to go in. And here is a list of the different projects we've done. It's a lot of information here, but you can see we've dealt with pretty much any target that anybody's thrown at us. Obviously, sometimes you run across cells or genes that are essential to the cells. Obviously, if that is known to be an essential gene, then we can talk about it, some strategies for that. In some cases, you could do a conditional knockout, or you could try and get a heterozygous where you have partial knockout of it. But in a lot of cases, if you try and do a straight knockout of an essential gene, cells do not like it, and it doesn't go well. So we've had some cases where that's happened, usually because it's not known to be an essential gene. But a lot of times it's just not a good direction to go in unless you have something that you're really interested in. But we have a lot of different approaches that we can take. We've done some more complicated. Obviously, a lot of these are just straight knockouts because they're pretty efficient to do at this point. We also do, some GFP tagging, introduction of specific mutations, some amino acid changes, things like that. And so far, we've done 61 knockout lines. This actually might not be accurate anymore. 5 conditional knockout cell lines, 20 gene knock-in cell lines, 11 gene tag cell lines, 8 knockout mice, 12 conditional knockout mice, 9 gene knock-in mice, and 10 gene tagged mice. And obviously, we're doing more projects every month, but we are always eager to try some new stuff. Talking about pricing a little bit, obviously the conditional knockout cell line with such a large construct to introduce is probably one of the trickier ones, just because you're removing a large piece and putting in a large piece back. So it's a lot tougher just because of the large template that can easily just be repaired quicker depending on how the cell is operating there. HDR mediated gene tagging, again, we're using homologous recombination, so it's just a little bit of a slower process. single amino acid knock-in incorporation, and single gene knockout obviously is the easiest. However, we're offering some discounts now for multiple gene knockouts that you might want to do. So if you're looking at multiple targets in the same cell line, or if you have the same gene and you want to knock it out in multiple cell lines, we can offer a discount just because a lot of times, if it's the same genome that you're working with, it's going to be about as efficient from cell line to cell line usually. Obviously, some cells behave differently than others, but we've been able to get pretty good efficiency doing the same knockout in multiple cell lines. So if you have multiple cell lines that you're interested in knocking out a gene in, talk to us and we can probably come up with something pretty efficient and useful for you guys. We also provide some support services if you're more comfortable with doing gene editing experiments. So using CRISPR-Cas9 guide validation. So we can order the guide, test it out, find the most efficient one, and then give you those results, and then you can do the editing yourself. Otherwise, we can, we'll provide those guides. Guide validation is a separate service, but not too much more expensive. And then we can also provide HDR donors. These can get a little bit tricky to produce depending on what gene you're talking about, but we'll provide that. And then, like I said, delivery of highly edited cells for your own subcloning purposes. We've done this a few times now, and so far people have been pretty happy. Because if you have, even like a 60% edited cell line, you're usually able to find some pretty good clones pretty quickly. Genotyping services is something that people use a lot, just because we have worked on the design of it, so we know how to get some decent primers for whatever it is, and we have a pretty good system for genotyping mice pretty quickly, just using puptos. So if our plate isn't too full, we can usually do some genotyping. If we have a free afternoon, if you deliver puptos that morning, we could at least generate PCR amplicons by that afternoon, send them in, get some results the next day. If it's a more complicated construct, obviously we have to do a little bit more genotyping, but it can be a way of kind of taking some of the burden of checking all of these individual mice off of your plate. And so that's something we're definitely very happy to offer, along with, guide RNA services and donor construction. If you have any comfort with it, obviously we can talk about different strategies. If you really want to go through and check one of these less efficient ones, you're definitely able to do that, and we can come up with pricing strategies for any scenario. But just talk to us about the project, and we're pretty happy to do that. So if you have any questions for us, obviously we'll talk about it now, but we also have, so in our lab, it's me, Chris Meiers, and then Summer back there. And then we got Brandon and Bo are our co-directors, and Walker is our interim coordinator. He's kind of handling a lot of the emails right now, which the email for that is just geser@umn.edu. So yeah, that's kind of us. Does anybody have any questions? Yeah.
I've got microphones.
Thank you for that presentation. First, I have two questions. First question, what about, you had human primary immortalized cells up there. What about human primary non-immortalized cells, like immune cell types,
I
etc.?
think we would probably have to talk about kind of the, kind of the robustness of the cell. It would probably be something that we can try, but we might just have to kind of I don't know, come up with a more, I don't know, specialized approach. Just because I know there are some that are, if your cells are not particularly robust, if you're using some of these editing methods, they can be a little bit problematic, and you get some cell die-off. And so if you have a large amount of cell die-off with these primary cells, then it can be tougher to find the ones that you want. So it's probably something we can discuss as as a group or whatever, and figure out a strategy for trying it out. But we do not have a huge track record of it, is what I would say.
Okay, but you're willing to try.
I think so, yeah.
Okay, and then I guess my other question is the conditional knockout with the LOX P sites is that's cool, and I'm looking sort of at the price tag and your comment that it's challenging. Is there a guarantee for those? Like,
yeah, so basically the way we work on it is, and we have had projects that have failed for one reason or another, but what we do essentially is we charge per phase of the project completed. So we charge for the design and the initial testing, and then we charge for subcloning, and then we charge when we have a delivered clone. So if we do not get something, then we would only charge a portion of the project, just for our time, basically. But at least for our conditional knockout cell lines, I don't think we've had a failed one of those. We've always been able to deliver something. It just usually takes a little bit more time, just because of the scenario there.
great, thank you.
What sort of price point are you envisioning for the iPSC editing?
That is a good question. I think it would be another scenario where we'd have to discuss it. Since we haven't done a complete project on it, I know the first one that we would do, we would be determining an estimate up front. I know the approaches we had talked about is either we'd be getting an estimate for media, because I know there's a specialized media that you have to use. The other approach would be if the lab is providing the media, since you have, if you have a specific cell that you're working with and the media that are good. We've done that with other labs where there's a specific media involved, so we don't charge for that. So I think we're still coming up with a pricing strategy. Obviously, it depends on what project you're working on, but we'd come up with a quote at the beginning so that we knew exactly how we would proceed. But I know we're planning on having like a specific iPSC engineering charge. We just haven't developed it yet.
Any other questions? All right, let's thank Chris one more time for a good seminar. Thank you.